This video is part of the lecture Plant Breeding in a Modern World in the scope of the course Applied Genetics at the University of Ostrava. Heredity is the passing of the traits from parents to offspring, which is made through the inheritance of genes. One of the most famous examples of heredity in plants is probably the heredity of traits in peas as studied by Mendel. Pea flower color has a fairly simple hereditary pattern. The color is determined by one gene with two different alleles, each corresponding to phenotypic variants, and with the complete dominance of one allele over the other, so that only the dominant purple allele is expressed in heterozygote individuals. Traits that have such simple heredity are easy to breed in order to obtain desired results. If you know the phenotype of the parents, you can easily predict the genotype and phenotype of all of their offspring. But other traits are not so simple to breed. Let's take the example of yield in wheat. The amount of the yield will vary from one offspring to the other, even if we knew the exact genotypes of the parents. Moreover, if the exact same genotypes were cloned and then grown in a different environment with more water and higher nutrient content in the soil, their yield will likely increase. This is because plant yield is a quantitative trait. It is determined by numerous genes, each contributing only a little bit to the final phenotype. And to make things more complicated, the expression of these genes often depends on the environment in which they're measured. So plants growing in fertilized soil would have higher yield than plants growing in nutrient-poor soil. It is not just plant yield that is a quantitative trait. Plant height, resistance to environmental stress, dormancy, phenology, and many other are quantitative traits as well. All quantitative traits have a continuous phenotypic variation in the population, which can be measured in units. They are controlled by multiple genes with small additive effects, and their expression depends on the environment. Improving quantitative traits via breeding might seem like a really complex task, but breeders have quite a few genetic tools at their disposal that could really facilitate their work. First and foremost, quantitative traits are not described as the typical Mendelian traits in discrete phenotypes, because this would be an impossibly complex task. Imagine a trait that is determined by 10 loci with 2 alleles per locus. There would be at least 50 possible phenotypes, and any 2 adjacent phenotypes differing by a single allele might be impossible to distinguish through phenotypic measurement. However, quantitative traits, as their name shows, are quantifiable. They can be measured in units, such as the number of seeds for the yield, meters for height, or grams for the weight. Because of this, quantitative traits can be easily described by their mean and variance in the population. We can represent quantitative trait distribution in a histogram, that is a bar plot in which the position of each bar corresponds to the trait value, here the weight of the carrot, and the height of the bar corresponds to the frequency of that value, namely the number of individuals in the population that have measured this exact value. The distribution of the vast majority of quantitative traits can be approximated by a bell-shaped curve, which is easily described with 2 parameters, the mean and the variance. The mean is simply the average value of the trait in a given population, and the variance is the dispersal of the observed phenotypes around this mean. Here is another distribution that has the same mean, but a smaller variance. This simply means that there are fewer carrots that are very heavy or very light. And this distribution has the same variance as the yellow one, but a higher mean, so on average, carrots from this group weigh more than the carrots from the yellow or the red group.